Welcome to Fertility Frontiers. I'm truly honored today to speak with two pioneers in the field of reproductive medicine and embryology, Professor Jeremy Thompson and Professor David Gardner, both co-founders of Fertilis, which is the company that we're going to be speaking about in this episode. So both of you have made key contributions that have shaped the way we understand and we practice embryology nowadays. Jeremy, could you please start by sharing a little bit about your background and your main focus on your research?
I've been basically working on embryology for 40 years, about that. And my interest has always been about behavior of embryos, oocytes, and how that relates to their developmental competence, and especially the environmental impacts that we apply to embryos as they're trying to grow, vitro culture, etc., oocyte maturation, in vitro, etc., and how that impacts their developmental competence in particular.
Oh, wonderful. Thank you for sharing, Jeremy. And David, I would like to hear the same for you, a little bit about your background and research interests.
Okay, I've come a convoluted journey. I started out in the UK with the great Henry Lees, and it was Henry who instilled in me a passion for the metabolism of embryos. And it was really by studying what embryos were doing metabolically did we start asking questions like, but why are we growing them like this? Because we're giving them things they don't really want. We're creating environments empirically that don't actually mirror what the embryo itself was needing. I was very fortunate to then go work with the great John Biggers at Harvard. And then I moved to Australia in the late 90s where I met my good friend Jeremy. So we've both been in this game for about over 40 years. And what an amazing ride it's been. When I moved to Australia, working with Alan Trounston and Carl Wood, I really wanted to try and do translational work. Like all of the amazing things we were doing in animal models, how does that relate to clinical IVF? And I guess that was a massive breakthrough. It was during that period in the sort of late 80s, early 90s, that Jeremy and our paths crossed for the first time, and one of many times. And I had the great pleasure not only of working on different animal models, but I got introduced to domestic animal models. And I spent time with Jeremy in New Zealand doing sheep embryo transfers and really understanding, as he articulated, that the environment that we grow the embryo in has a profound effect on downstream consequences. And that led me to work more and more in clinical IVF. But my passion has always been understanding what makes embryos tick. And then how can we translate that into better culture environments, culture conditions? And at the back of our mind, Jeremy and I would always scratch in our heads saying, but what we're doing in vitro is so unrelated to what our gametes and embryos see in vivo. And I think it was then that we started, way back in 93, Jeremy, I think I was in New Zealand with you, and we started talking about how can we better create systems that could help us grow embryos. And that, I think, was the genesis of where we are today. Because even back then, 30 odd years ago, we started talking about how do we grow embryos in a dynamic environment? In other words, how do we perfuse them? How do we grow them? So the technology didn't exist back then to do the kind of things we wanted to do. But today's a very different day.
I'm actually super excited about learning a little bit more about the platform that you're developing. But if it's okay with you, I want to throw some rapid fire questions for you to answer in short answers, like 10 seconds or less, to get to know you a little bit more outside of your scientific lives. So let's start with two easy ones. Jeremy, early riser or night thinker?
A little bit of both. It depends on my health condition at the time. Yeah. I generally probably prefer early.
Good. I'm the same. David, coffee or tea?
Coffee. And before dawn, I'm out of that bed.
I'm the same, but because I have a 1-year-old, it's not my choice, really. Jeremy, what is the most exciting moment in your research journey so far?
Oh, it definitely would be surrounding the product we've been developing. The most exciting moment was when I saw a 1 millimeter Eiffel Tower that had been created by a process called 2-photon polymerization. And that gave me that sort of shivery moment and saying, if we could do this 1 millimeter Eiffel Tower, if we can construct that, we can construct devices for embryos at their level of size.
This is an awesome answer. And it reminds me, 2 days ago, I was speaking with Jack Cohen, and I asked him, when do you think that the best opportunities arise. And he said when two different technologies converge, and it's exactly what you were explaining here.
Yeah.
So, David, if you weren't in science, what career would you pursue?
I would be a musician.
Yeah.
How's
Nice
that?
one.
He's a great musician.
I'm a frustrated musician who uses science to pay for my hobby.
It's true. I remember seeing you playing the guitar in one of the Future Life events. Very good musician. So, Jeremy, define Fertilis in one word.
In one word, revolutionary.
Good one. David, one piece of advice for young embryologists.
Dream big. Dream big. Never take no for an answer. Just because you can't do something today doesn't mean to say you won't be able to do it tomorrow. So just keep going.
Good. And the last one for the two of you, but let's start with Jeremy. What is, in your opinion, the biggest challenge in IVF right now?
Minimizing the impact of variable environments created not only by the conditions that they're cultured in, but also the variation in the different procedures that are conducted.
Would you agree, David? Is that, in your opinion, the biggest challenge?
I agree with everything Jeremy said, but I would add the difference now compared to where we were 30 years ago is the sheer volume of the patients we're trying to treat. As taking everything that Jeremy said, but apply it to a clinic. So 30 years ago, a clinic was 400 cycles. Today, they're 4,000 cycles. So being able to apply that ethos to a huge volume demands a change in how we do things. And I think this is part of where we were coming from in the whole fertility arena, really, is rethinking how do we do what we do to accommodate the principles that Jeremy just articulated.
Sure. So that actually leads me to the first question, because I want to understand a little bit more of what you're doing at Fertilis, which is what is the specific challenge or problem in the IVF field that you're trying to solve, and how are you trying to tackle it?
From my point of view, we're just trying to make the embryology laboratory more efficient, user-friendly. The amount of knowledge and expertise that embryologists have to have is extraordinary. And yet, and you may have a different opinion, but a lot of the work that gets done inside an embryology laboratory is routine. Now, those two, when you have complexity and a large need for being able to establish technology and techniques in a laboratory setting, and yet it's done routinely, this leads to what people would describe as automation. Because if you've got something complex, but it's done on a routine basis, it's ripe for automation. Now, we're not necessarily automating steps themselves. What we are enabling the reduction in expertise that is required for significant steps within the IVF pathway.
And David, with the platform, the technology that you're developing at Fertilis, what are the specific procedures that you can do at this time?
I think the reality is IVF really hasn't changed that much. And I think where Jeremy and I came in was looking at the processes that we've been doing for 30, 40 years and saying, is that really now the best way to do that? And look at it with fresh eyes. So I think one of the things that we started with was something that our challenge was, could we create a device to perfuse the embryo? And came across that. And I think we had a proof of principle paper that really shows that you can do it now. But that's again through the 3D printing technology. But what came to us as we were doing that was that it was so versatile that if we could come up with another idea, we could pivot very quickly and apply the technologies to that. And the first thing that really came up was ICSI. When you look at how we've done ICSI since Jean-Pierre did that, the first paper all those years ago, we still use a holding pipette and we still use an injection pipette. But when you watch ICSI being done, or you see the constraints and the pressure being put on the egg on that holding pipette, and you have to do one egg at a time. So we came up with this idea of, couldn't we create an array? And rather than have a suction pipette, we could have a holding device, almost like an egg cup, a 3-dimensional egg cup, but have a whole array of egg cups. It's just a nice way of visualizing it. So the egg cup itself then absorbs the pressure when the needle goes in. And it would facilitate, as Jeremy alluded to, automation to some degree but it certainly would make doing an ICSI a lot more streamlined.
So at this point, with the devices that you're using, you can replicate or you can perform ICSI and also embryo culture. Is that correct?
So our first product that we generated is ICSI dish, and there is an insert within the ICSI dish that is this array arrangement that is created by this 2-photon polymerization process. The idea of doing a microfluidic device using the same technology we have established, but it is not in a commercial pathway as yet.
But I think what's really key is that we've done the proof of principle. We can actually do it. Example, we tried to do perfusion culture independently for many decades, really, using microfluidic devices that were available to us, but they really weren't sufficient using that kind of technology, using PDMS and self-lithography. With the 3D printing, it was a complete game changer. And the ability to create new devices, optimize, refine, you could never do with something previously to this. It really is a remarkable situation we find ourselves.
To perform the proof of concept, have you been using an animal model?
Yeah, we've been using primarily the mouse model, but we also have worked with cattle, sheep as well, and pig, to provide the animal data to support our concepts.
And what is your pipeline? Are you trying to develop the device and prove its efficiency and safety to be able to be used at IVF clinics, or is for the animal field?
So if we're talking about our micro-ICSI device, which is our first product, yeah, I mean, we've taken it all the way through to clinical evaluation and in the foundational clinical trials that are starting very shortly. A very exciting period for us, and Dave and several other entities around the world are heavily involved in those clinical elements. We have a pipeline of devices that we are working towards. We have a biopsy device that we have created, which we haven't started to evaluate yet, but the design has been created and is shown to be working, but we haven't gone any further. And then, as David has also alluded to, we have a microfluidic concept that we have publication on, but as yet it's no further developed beyond research.
And so you already have an approved study to be used in an IVF clinic. Are you going to perform this in Australia?
Yep. That study's about to start right here in Melbourne, actually. But we've also got proof of principle data on fertilization from colleagues in Japan. So that with all the extensive animal studies, then we have proof of principle data for fertilization in the human. We know it works. And now we're going to do a proper full study here in Australia, which is tremendously exciting. And to see how quickly we've been able to get from one part to the next it's great.
I think the most exciting aspect for us is that whenever we've taken it to clinic for even demonstration purposes, no matter whether it's Australia, Japan, etc., everybody who uses it says, this is great and we need it in our lab now. Yeah.
Do you happen to have the device around you that you could show to the camera?
Yeah.
I'm so curious.
I could briefly share the screen just to show you what it looks like.
Yes, please do.
Thank
Yes.
you. Thank you, David, because I would be challenged technically to be able to do that. I'm a bit of a dinosaur when it comes to.
Oh, I'm not far behind you, mate. You just had the paper open so I could see it. So you can see this from our paper. This is from jar. And the key thing is it's here. This is the array. And this is what it looks like in close up. So the egg sits right here. And the ICSI needle, the sperm are in here. This is your PVP, your sperm, your PVP. You pick them up. And you can just do the injections in sequential eggs as you need to do. So they're in groups of 5. So there's this array is a 20, but all of them are numbered. So it's very elegant and time saving.
There. And there's a little circle on the left-hand side above a slit. And that's actually a number. And that number would be about 3 or 4 microns in size.
Wow. It's unbelievable.
Yeah, I'm trying to get a little bit bigger. There we go. Just here.
Yep.
Yeah. Incredible. So the egg just sits here, yeah.
I can imagine that to develop this, you have involved a very multidisciplinary team of people, not only embryologists, right?
Yes.
So who is part of your
Yes,
team?
requires engineers and specific types of engineers, computer assisted design, as well as the capability of understanding how this light-based polymerization process occurs.
How your technology compares to what others are developing, for example, Conceivable or Overture?
That's a great question. We see what we're doing is very complementary to what they're doing. I guess what we would argue is that Conceivable, for example, has recapitulated what an embryologist does, but through robotics instead of a human. What we're saying is that can we find better ways for enabling the work of an embryologist to be performed?
Yeah.
So is it simpler? Is it easier for them? Does it take the stress out? De-stresses the embryologist? So that's our philosophy. Whereas Conceivable's philosophy is that we can replace the embryologist with robots. Now, the two are actually complementary, and we believe that we could be working together.
Yeah. But I think what Jeremy said is, I like that de-stressing. One of the things we've talked about for many years is how important it is to alleviate stress on the embryos to facilitate them to grow properly. And yet, time, we know that being a clinical embryologist is an insanely stressful job. Physically, it's very demanding. So we couldn't understate the impact of the daily ergonomics of IVF to the people using these technologies. And I agree that Jeremy's right. This tees up very nicely for automation, but it's also de-stressing the actual workflow for the embryologist.
What are your thoughts? This make me wonder because the first device that you're developing, or the one that is more advanced, as I understand, is for the ICSI. And I'm still in my mind all the time wondering, why are we not using more conventional IVF versus ICSI, which is more invasive? Are you trying to also work on conventional IVF?
I do believe that in the future, our device, our end product, will look like a platform of several workstations. One is an ICSI workstation, but on the one dish. But there will be an IVF workstation as well, but it'll all be on the one dish. It'll be a pretty big dish, but it'll be all on the one dish.
Yeah.
And if you could give me a timeline, the way you picture it right now, on when are you going to be finished with the clinical trial that you're doing, and when this would be potentially available for the clinics to use?
Effectively, it'll be in the next 12 months, we'll have a complete array of data from several sites around the world.
Tell me a little bit more about the business plan. So you would be producing the devices. Can they be used with any microscope, or it's only specific brands? Would you be selling globally? What is the plan that you have in mind?
Effectively, our business plan is to create a dish that has different workstations that allows you to do pretty well much all the major components of the steps through IVF. That's the vision. Okay. As a business plan, we are doing it as an iterative process. So we're developing effectively one capacity, and then develop the next capacity, add it to the dish, develop a further capacity, add it to the dish. So it's an iterative process.
And with regard to this device, for example, the micro-ICSI, it's compatible with any microscope, any workstation at all. It's not specific. So that's the beauty of it. It's, as Jeremy said, we printed into an ICSI dish. Where you do ICSI, you can just use the device.
Wonderful. This is really exciting, guys. Before we finish with the interview, I wanted to use the opportunity that I have, the two of you here. And it's, I think, a great chance because you have an overview of the last 4 decades in our field. So, David, since you developed the Gardner grade system, and since then, there's been many other tools developed for embryo selection, like GTA. From your perspective, what do you consider the best approach today for selecting the most viable embryo?
To date, in all humility, how we use it today is the Gardner grade. We just published at the end of last year a prospective randomized trial comparing artificial intelligence to that grading system. and the grading system comes out on top. And I think the reason for that is because it has so much physiology built into it. It wasn't something that I just came up with. There was method in the madness. It's quite a nice story. And so once you look at that, and then when you look at the studies that are coming out on AI and say which parts of the AI are heavily weighted, it is blastocyst expansion, it's trophectoderm, and of course it's the inner cell mass. And those are the essence of that grading system. Now, the only downside, of course, is it's subjective. I always say, when is a 4AA blastocyst not a 4AA? And that's when somebody else grades it. I also remember when Marcus Montag created an online grading system and he asked me to test it. And I failed. And I just went, wait a minute, I failed my own grading system test. But it comes out that AI will come along because it will at least standardize that grading. And of course, it's quicker. I'm very happy that the grading system lasted so long, but I expect it to be superseded by something soon. Yeah.
I'd like to add that I think these new thinking around non-invasive technologies to assess embryo metabolism. After all, we are talking to the person who has done the most on metabolism of the embryo and has shown conclusively that the metabolism of the embryo is associated with viability, subsequent viability. The incorporation of artificial intelligence along with non-invasive metabolic assays is going to be the way in which this story ends.
Totally. And I think the reality is that we will get so much information about the health of the embryo. And Jeremy alluded to the fact of our work over the decades has also been how the embryo interacts with its environment. And one of the key papers that the two of us published together in 95 was to show that actually the environment that, in this case, it was a sheep embryo, can actually impact fetal development downstream. And we're getting to the point where it's coming to the AI, but totally agree, mate, with the metabolic assessment. It's not just about viability. It's actually about technically health span of the individual as a result of that consent in vitro.
But it'll be the merging, the converging of these technologies. It won't be just AI alone. It won't be just metabolism alone. It will be the convergence of several technologies that will be the end.
You have mentioned AI that is going to potentially use the morphology as guidance, right? Like the vision of it. And metabolomics. Do you see the value in testing the DNA of the embryo through a biopsy or non-invasively?
There is work being done in Adelaide around and other centres that is looking at the relationship between metabolism and DNA and others.
And if we're talking about cell-free DNA, again, as a reflection on the ploidy alone, I think perhaps not in the near future, but in combination with AI assessment of morphokinetics, in combination with the metabolic profile. And in fact, we've just submitted an abstract to ASRM in which we looked at aneuploidy in human blastocysts and their metabolism, and they're different, which is consistent with the lovely data out of Denny Sakis' lab in Boston and Kylie Dunning's lab in Adelaide. So when we look at the culture media, sample that, look at the glucose metabolism, it's different in the two types. So you have all of these wonderful independent variables. You put that together to create an algorithm. I think, yes, then Jeremy and I have discussed this, the days of potentially embryo biopsy per se are numbered. We'll replace it with a multifaceted approach.
That's really interesting. And as of right now, and until we develop that technology, do you see justification for doing an invasive biopsy in an embryo or not really?
I think we both agree that biopsy is place still at this present time. We're certainly devising a device that makes biopsy easier, simpler, etc. But no, I agree with David that one day the convergence of these other technologies to describe what a good embryo is with normal ploidy status, or at least acceptable ploidy status that will lead to an ongoing pregnancy, will emerge.
But until then, yes, I do think we should be doing the biopsy. Certainly for advanced maternal age, recurrent implantation failures, there's a whole group of patients who would benefit and will benefit from that approach. It's just going to be wonderful if we don't actually have to do the physical biopsy. You can replace it with some of these other alternatives. Yeah.
And Jeremy, I wanted to ask you something. In my personal experience, I started working in an IVF clinic and I spent most of my time working with human embryos, but I did my PhD with the bovine model with Beatriz, and Pat Lonergan.
I know
And
Demetrios very well.
I realized how much knowledge there is already in the animal field in reproduction that we're not really getting in the human field. Maybe because we're not putting the time in reading the papers. Like, why is there such a disconnect?
That is an interesting question, because I would have said about a decade ago or even 15 years ago, I would have thought that animal embryology, livestock embryology, was ahead of what was happening in the human IVF area. It's reversed now. The use of artificial intelligence, the degree of manipulation skills that embryologists have in human IVF, and also development of the assays such as what we've been describing that Denny Sarkis and Kylie Dunning have been doing. Livestock embryology has actually fallen behind, and it's now up to them to catch up.
Did you have anything to add to that, David?
I think Jeremy's right. I think I was very lucky that in my career, I started out in the mouse, then moved into the goat, the sheep, the human, even the wallaby and the cow. I worked on a whole variety of different species. But going to those different conferences back in the 90s and the noughties, it was really amazing how much data that we did get out of the animal models that, as you alluded to, sadly, a lot of people coming into human IVF are not aware of. I also agree that with Jeremy, though, that in the last few years, it has flipped. Technologies have been invested into human IVF at a great rate, which is tremendously exciting, though. But it's taken all of those things to make us, to get to where we are today, though, for sure.
True. I truly cannot thank you enough, the two of you, for being here. For me, it's an immense honor. I honestly started this as the teeniest, tiniest platform and still is. And I appreciate you putting your time and believing in this small project. And yeah, thank you. Really, thank you.
Thank you.
It's been a very enjoyable half hour. Thank you very much.